So we're going to start our discussion of muscle today. Now I know that you are deep into the muscles in the lab, naming the muscles. You're doing origins, actions, and insertions. Y'all doing that in the lab? So what we're going to focus on is more of the cellular aspect of muscle. And again, you probably saw some of this with your histology as you did at the beginning of the semester. But we're going to first look at one of our three types of muscle. Now, what are our three types of muscle? Skeletal muscle, cardiac muscle, and smooth muscle. Almost in this order. Skeletal, cardiac, and smooth. Now, for the most part, cardiac and skeletal muscle are going to be fairly similar. Their physiology and the way they contract is going to be practically identical. So we're going to start with skeletal, but really sort of compare these two closely. We'll talk about some of the differences in cardiac and then finish our muscle unit with a discussion of smooth muscle that's the most different. And how it contracts is pretty different as well. But the first thing we want to compare are muscle cells in general, how they are different from just your generic cell in your body. And the first difference we're going to see is we put a whole bunch of prefixes onto everything called sarco. Now, in lab, if you had to say what the prefix for muscle was, you probably wouldn't say sarco. What would be the prefix for muscle that you hear a lot? Myo. That myo prefix typically refers to a gross anatomical level or even a histological level. But when you get to the cellular level and we refer to muscle, that's where we use the term sarco. And the differences we're going to look at in our muscle cells deal with the plasma membrane differences, structures, and the abundance of certain proteins are certainly unique to muscle. That's going to be in the cytoplasm. And then we're going to have a very highly specialized endoplasmic reticulum. Now, it's a modified smooth ER. We know that the rough ER was principally functioning with protein production for secreted and membrane proteins. What did the smooth ER do in most of our cells? What did it make? Lipids, hydrocarbons. Well, for our muscle cells, skeletal and cardiac, we're going to see that the sarcoplasmic reticulum deals with calcium. Write that down somewhere. We're going to see it in another couple of slides, but go ahead and begin to wrap your brain around the fact that the sarcoplasmic reticulum deals with calcium. Rough ER, proteins. Smooth ER, lipids. Sarcoplasmic reticulum, calcium. And that's going to be key to how muscles function. Calcium plays a tremendous role in what we do. Now we'll start with the plasma membrane. And not unlike what we've seen in other cells, phospholipid bilayer, we've got our membrane proteins, we've got plenty of sugars on that. But the thing we're really going to focus on in the difference is the semi-permeable nature of the muscle membrane. Now what does semi-permeable mean? Is semi-permeable a brick wall? No. Some stuff can pass, but not everything. And so like we see in other cells, small, nonpolar molecules, they can simply pass through the phospholipid bilayer. What is that transport process? Simple diffusion. But in muscle cells, we're going to regulate the transport of a lot more than simply these types of molecules. And so we're going to have a lot of membrane proteins that are functioning, not the least of which are channels that are referred to as leak channels. We're going to have gated channels, and we're going to spend quite a bit of time talking about these today and tomorrow. And then we can also have carriers. Now, these are still transporting material still from high concentration to low concentration. So what's that transport mechanism? Facilitated diffusion. And in fact, we're going to see quite a bit of this happening. And we'll draw your attention to those as we get there. Now, I'll go ahead and introduce the fact. And we've talked about it since the first unit. What is going to be critical for you to understand? Not only is calcium, that ion, going to be important in a contraction, present in the sarcoplasmic reticulum, but we are going to be using, in almost every step of a muscle contraction, the ions sodium and potassium. We've talked about that since the first unit. And in the first unit, I said, go ahead and commit to memory. Where is sodium in highest concentration relative to the membrane? Outside. Now, what is our relationship between sodium and potassium? Opposite. Remember? Opposite. So if sodium is high outside, where is potassium high? Inside. You need to start recalling that. Now, one of the main reasons, especially for our muscle cells, that sodium is highest outside is because we have a lot of this in our plasma membrane. There you can see we're using energy, transporting against our gradient. 3 sodium out, 2 potassium in. Who's that? That's the sodium-potassium trunk. Pump. What in the world am I trying to say? I was trying to say active transport too. Sodium-potassium pump, and that is our active transport mechanism. So we're going to be creating concentration gradients. We're going to be allowing the movement of ions based on their concentration gradients. And all of that is going to play a role in the signaling of our muscle cell to contract. Now, a lot of what we're going to talk about with our channels, ions moving, sodium, potassium pump, we're going to be able to directly relate that to nerve cells in the next chapter. So it's not that we're going to learn this about muscle and just forget it. This is going to relate to both. So we're going to walk through that pretty carefully, and we'll probably get to the bulk of that signaling on Wednesday. Now, we're going to spend a little bit of time here because we talked about the sodium-potassium pump, moving 3 sodium out and 2 potassium in. What charge does sodium have? Positive charge. You had 50-50 shot, right? Positive charge. Now, this is the first time that sodium and potassium are opposite. What charge does potassium have? It's also positive. So as we're pumping out sodium and pumping in potassium, we're affecting the charges inside and outside of the cell. Because if we're pumping out 3 positive charges and only bringing in 2, kind of seems like the outside of the cell might be more positive than the inside. And in fact, that's the case when we look at most cells, and especially in muscle cells. And so this separation of charge, and that's what our semi-permeable membrane is doing, it's going to create a voltage. Much like the battery in your remote control. Are there 2 positive ends of that thing? No, there's a positive and a negative. It's polarized. It has a voltage. And so your plasma membrane, with this separation of charges, creates a voltage. And typically the inside of the cell is going to have more negative charges. And so this charge difference we call a potential. You know, we talked about potential energy. Well, potential energy means you can get work done. And right now with a charge separation, there's the potential to move that, change that voltage. And so when our cell is doing nothing, we say that our resting membrane potential. And for muscle cells, that's going to be in the neighborhood of negative-95. When we get to neurons, that number is going to be negative-70. And in fact, negative-70 is a number that I like to think of for resting membrane potential. So if you want to change that to negative-70, that's cool with me. I like numbers with zeros on the end because I can do math easy with zeros and 5s. So since we're talking about voltages, the little m is milli. millivolts. So this is negative 95 or negative 70 millivolts. The number isn't so critical for me, but that negative number is. Because that negative means the inside of the cell is more negative than the outside. If that was a positive 95, what would that tell you about the inside of the cell? It's more positive than the outside. If that number was 0, what would that tell you? It's neutral. There's no charge difference. So negative millivolts means the inside of the cell is more negative. Now, let's look at a difference here. Why is the inside more negative? There are a couple of different reasons. We've already touched on this one. The sodium-potassium pump. It pumps 3 positives out, that being sodium, and it pumps 2 positives in, that being potassium. So if you're pushing more positives out than you're bringing in, certainly that's going to contribute to more positive charges being outside. See, that's a way you can increase the negative on the inside, by getting the positives out rather than having to bring in more negatives. So our sodium-potassium pump plays a critical role in doing that. Of course, it's going to take a lot of energy to do that. But the other thing that I want you to pay attention to here, we're kind of focused on charge right now, but I also want you to keep up with the concentration gradients. Because what do you see up there at the top? High what? High sodium outside. What's down at the bottom? High potassium inside. Now with our muscle cell, one of the ways we're going to signal is by opening specific channels that will allow sodium to undergo facilitated diffusion and other channels that are going to allow potassium to undergo facilitated diffusion. Now remember, diffusion is downhill. And if you want to go really, really fast on your bicycle, are you going to find a really slow gradient, or are you going to want a steep gradient? And if you want to go as fast as you can, you're just going to take your bike and go skydiving, right? That's just straight down. These concentration gradients for your muscle cell are practically straight down. And in fact, you may have 150 units of sodium outside and 5 inside. And the reverse is positive for potassium, maybe 145 to 10. So it is an extremely steep gradient. That's so when your muscle gets a signal to contract, those ions change place, it signals rapidly and the contraction can happen rapidly. Because when you touch that hot stove, how long do you have to move your hand? Not long before you're getting first, second degree burns. So that's why these signals have to go rapidly. Now we're going to talk about the signaling again Wednesday, but I want you to understand the significance of why we're talking about these now. We're putting the pieces together, and it's probably not all going to make sense until Wednesday or Friday. But just kind of hang in there with me. I want to make sure that I don't lose anyone, though. So if you're not kind of following along with what we're doing, stick your hand up and let's get caught up all together.
Do they use up the sodium and potassium, or they get?
No. Whenever they change place, sodium-potassium pump puts them back. Sodium-potassium pump is constantly working. That's why, what was that number? 12% of all of our energy in our body is used by the sodium-potassium pump to maintain gradients. Not just in our muscles, but also in our nervous system. Because the nerves are doing the same thing. So sodium-potassium pump, helping create the gradient. The other reason that the inside is more negative is we have these things called leak channels. Now, when I have a leak, I have to fix it. You know that leaky faucet that drips all night? You just can't sleep with all that dripping? But for some reason, evolution has decided to keep these leaky potassium channels. And my hypothesis is the leaking potassium keeps the sodium-potassium pump turned on to maintain that steep gradient. Maybe without these leak channels, this would shut down and the gradient would sort of soften out a little bit. I don't know. That's just an educated guess. But regardless, muscle cells in their plasma membrane have potassium leak channels. And as these positives leak out, it helps to create a more negative on the inside. So the pump and our potassium leak channels, two of the reasons that the inside is more negative, with really a third reason, and possibly one of the most important. If you want to go ahead and put reason 3 by this, you can. And highlight it. But when we look at ions moving and charged molecules moving, only those things that can move across the plasma membrane affect the potential. They affect the change of potential. DNA. Is DNA ever going to be transported out of the cell? You hope not. That's a dead cell if it does. DNA has a lot of phosphates. That's that sugar phosphate backbone. What charge do phosphates have? Negative charge. You have a lot of cytoskeletal proteins that have negative amino acids. Are those cytoskeletal proteins ever going to get out of the cell? We hope not. So for all of those fixed anions, fixed negative charges inside the cell, that more than anything else is going to contribute to our negative resting membrane potential. So really, these 3 criteria play a tremendous role in the inside of our cell being more negative than the outside. Now understand, when we say more negative inside than out, understand that's like a gun that's loaded, just ready to be fired. That's a cell at rest. And when the firing of the gun is going to be our muscle contraction. All right? Everybody, are we all up to date on this? We good? Because again, it gets more technical and more technical as we go. All right. So when we signal the muscle cell, this is going to happen from a nerve. And the nerve is going to secrete some chemical signals. That's going to cause our sodium channels and our potassium channels to open, and these ions change place, and it's going to flip our membrane potential. It's going to flip it from more negative inside to more positive inside in an instant. And when you graph this change out, it's going to look like a wave. And this wave is going to flow across the plasma membrane of our cells. Now, have you ever been at a football game? I mean, a big football game. There wasn't many people here for homecoming, but in a big, humongous, real stadium where people did the wave, people are standing up, sitting down, standing up, sitting down, and you can see that standing up wave just going around the stadium. That's essentially what's happening with our change of potential across the membrane of our cell. And it's simply the movement of sodium and potassium. But that wave of electrical activity is going to stimulate the muscle to contract. And so if we want that wave to move more rapidly across the cell and throughout the cell to cause the muscle to contract more completely and more rapidly, what can we do to our membrane that will help that happen, much like we did to our intestines to help us absorb more material quickly? More surface area, more membrane. We're not going to make the cell bigger, but the way nature has chosen to do this is by invaginating the plasma membrane down throughout the cell in these membrane tunnels that are referred to as T or transverse tubules. When you were a kid, you probably got the balloon and you stuck your finger in it, and you could see your finger wrapped with a balloon on the inside. Well, this is like a finger got stuck in and the finger got removed, leaving that tube of plasma membrane going all throughout the cell. And so if you looked at the surface of a muscle cell, it might look like Swiss cheese with these T tubules going all through it. So our voltage change, that wave of voltage change as it's flowing along the plasma membrane, it's going to flow down through that T tubule. to spread the signal more rapidly and completely throughout the cell. So this is another difference that we typically don't see in many of our other cells. Now, our modified smooth ER, the sarcoplasmic reticulum, which we've got illustrated in blue here, and just reticulum means network. And you can see how this looks like a netting all around our contractile proteins that we'll get to in a bit. But our sarcoplasmic reticulum, as it gets close to our transverse tubule, see how this is a lot wider and a lot bigger? The ends of our sarcoplasmic reticulum contact the walls of the T tubule. They have receptors that are shared. And so as that electrical wave comes down the membrane and comes down the T tubule, that electrical change is going to trigger receptors not only in our transverse tubule membrane, but in our sarcoplasmic reticulum membrane. And what does sarcoplasmic reticulum do? What's that ion I told you to remember? Calcium. The sarcoplasmic reticulum stores calcium. And when that electrical potential slides down, that electrical change is going to trigger the release of calcium from the sarcoplasmic reticulum. And you can basically look at the concentration gradient as sarcoplasmic reticulum 1,000 inside, cytoplasm 0. So when that electrical trigger opens those calcium gates, there is going to be a tsunami of calcium that is going to flow throughout the cytoplasm of that muscle cell in every place where a T tubule contacts the terminal cisternae. That's going to spread the calcium throughout that muscle cell in an instant, as soon as the signaling happens. Because we've already said calcium is important, not just for bones. And we're going to see how essential that is. Now understand, we haven't contracted the muscle yet. Muscle still hasn't contracted. We picked up the signal from the nerve. We transported the signal across our membrane down into the T tubules that triggered the release of calcium, but we haven't even contracted yet. We're still setting the stage for that to happen. All right? Now, let's move away from the membrane and look at the stuff in the cytoplasm now. We talked about the membrane. We talked about the sarcoplasmic reticulum. But principally, most of our muscle cell are going to be comprised of these contractile proteins that are arranged in these cables called myofibrils. Now, notice, we've got a myo, which means muscle, fibril, and we've got many of these cables inside the plasma membrane of one muscle cell. And what do we call that muscle cell? A muscle fiber or a myofiber. You see how these are sounding very much alike? Again, this terminology gets real close, but you've got to keep it distinct. A fiber is a cell. A fibril is the organelle and the bundle of our contractile proteins. And these bundles of our contractile proteins are called filaments. And the filaments that we have, starting today and throughout the rest of the semester and your life, because even if I'm talking to you when you're 50 years old, if I'm still alive, and you say the words without thick or thin, I'll go back and take points off. That's how important it is to always, you know when you're in trouble with your parents? How do you know when you're in trouble? When you hear all your names, right? So these are always going to be in trouble. We're going to use both of their names. We're not going to use all three because that's time to just leave home when you hear all three of your names. So our thick and thin filaments, they're going to be arranged in an overlapping pattern that's going to give that banding look. What did we call banding of our muscle in lab? Not striped. What's that word? Striated. And it's going to be the overlapping nature of these thick and thin filaments that give that striated pattern to skeletal as well as cardiac muscle. These thick and thin filaments are simply arranged differently in smooth muscle where we don't see that banding pattern. But if you don't have thick and thin filaments, you will not contract. So smooth muscle still has them as well. So if we could look more closely at one of our filaments, you would see in the middle of this illustration our thick fuzzy filaments. And you can see how the thin filaments have overlapped them. And I'll go ahead and give you a preview of a contraction. Our thick and thin filaments are going to interact with each other. And these thick filaments, if you can imagine my body being right in the middle of that thick filament, and we're going to use our arms as analogies to our thick filaments. We're going to grab the thin filaments, and by using ATP, we're going to pull them toward the middle of that thick filament. And do you see how it's going to shorten that unit we're looking at as we pull in both directions? And we're going to have these lined up one after another so that in these little microscopic units, they contract a little bit. But when you've got them linked in series, that's going to cause a muscle to contract possibly by centimeters. All because of these little molecular interactions and this minuscule contraction of each one of these units. So, myofibril. Here are our thick and thin filaments. Do you see why we call them thick and thin? I mean, look, these thick guys, they've got all these little double heads sticking out on them. And the thin filament are just these little twisted, beaded necklaces. Now, we've got some associated proteins here with our thin filaments that we'll have to consider. But we're going to consider these next. Let's look at the thick guys first. So our thick filaments are myosin. So from now on, you're going to say thick myosin. Not only is that going to help you with its function, but it's going to help you understand its arrangement within our contractile unit. Now, we call it thick myosin because on our filament, you may have 300 of these myosin molecules. And each one of those 300 is actually a doublet, two myosins that are twisted together. They have a tail region and they have a globular head region. And when we look at that individual myosin molecule, in many aspects, a lot of textbooks, a lot of biologists, they will use the analogy of a golf club to our myosin molecule. Do you see how that looks kind of like a golf club? Okay, I don't want you to do that. I want you to think of the myosin molecule as your arm, where the globular head is your hand. Now, one reason I want you to think that is because our myosin molecule is going to have two hinge points. Our myosin molecule will bend like it's your wrist. But the myosin molecule is also going to bend like it's your elbow. Golf clubs only have that one bend, right? Unless you play with my good friend from college who shanked a tee shot and bent his shaft over his knee. I didn't have that kind of money in college. I don't know where he got his golf clubs from. Yeah, golf clubs don't usually bend like that. So that's why we're going to use our arm. Now, notice on our globular head, there's two spots. And this is the other reason why I like to use the arm as an analogy. What are your hands good at doing? Grabbing stuff. And the stuff that the myosin hand is going to grab, first, it's going to grab energy. And so what do we need to grab? ATP. And as soon as it grabs a molecule of ATP, ATP breaks down into ADP and phosphate. And the energy is released. But it's all stored inside the hand. Nothing is released. The energy is there, the phosphate is there, and the ADP is there. It's preloaded. It's cocked. It's ready to go. But the other thing that the myosin hand can grab is a thin filament that we call actin. Actin thin filament. So I think you can already see where we're going, right? As far as a contraction goes. Here, this midline right down the middle of our thick filament. Just imagine that's my body. These are my arms. You can see my hands. Myosin hands grab actin on either side, and they pull everything towards that middle line, and that's going to pull each one of these units closer together. Can you see that already? That's where we're going with the contraction. Now, we're putting all the pieces in place, much like we did with everything else, like we did with transcription, translation. And next time before we get into, or maybe Friday before we get into the actual contraction, I want to show a video that will then go step by step by step. And hopefully by the end of the week, we will all be on the same page of understanding not just all the components, but how they work together to get a muscle contraction. All right? So thin filaments. Our thin filaments are principally going to be composed of the major portion that is going to be our actin. Now, these were popular back in the 80s. So maybe your mothers had these. Does anybody remember their moms having the twist-a-bead necklaces? There were these beads, and you could buy all different color beads, and you could twist them together. And some, it seems like in Texas everything has to be bigger. So some people had these big, humongous, like 50 beads twisted together, kind of like the homecoming mums that have to be bigger than the person now. But here are two beaded strands that you see in red. Those are the actin molecules. Now, each strand is called a filamentous actin, and each one of the beads is called G-actin or globular actin. And just like a bead that has a hole in it to string the thread through, notice that there's a little black spot on each one of those G-actin molecules. That little spot that you see is a myosin binding site. It's a myosin handle where myosin can reach up and grab the actin so it can pull it and slide it during a contraction. Now that's really all you need for a contraction to happen. Myosin and actin and energy getting together. That's it. But we also have these other molecules, not the least of which is this green thread that's running along the groove of our filamentous actin. And note what's happening here with this green thread. It's sitting over those myosin binding sites. And that green thread is called tropomyosin. Now, this is a tricky one, right? Tropomyosin. Is it sitting on myosin or is it sitting on actin? It's on actin. So why in the world we call it tropomyosin? Well, it's one of those names that you just have to remember. Because at rest, when the muscle is relaxed, tropomyosin prevents myosin grabbing actin. Because let me go ahead and preview this for you. If myosin has ATP, and if myosin can bind to actin, you will have a contraction. Did everyone hear me? Listen carefully. I'll say it again. As long as you have ATP, and if myosin can bind actin, you will get a contraction. So the only way to relax the muscle is to prevent myosin binding to actin, and that's prevented by tropomyosin. When it gets in the way and myosin can't bind, your muscle is relaxed. Well, that creates a problem then, doesn't it? Because to get a contraction, you got to be able to have myosin bind to actin. So to get a contraction, what do we have to do with tropomyosin? We got to get it out of the way, right? Moving tropomyosin out of the way so that a muscle can contract is the job of this series of proteins called troponin. Now, troponin is a protein that has a quaternary structure. Do you remember quaternary? Separate proteins together that make up a molecule. And troponin has 3 separate proteins that work together. One of the proteins of troponin binds to tropomyosin. Another of the proteins of troponin binds to actin. And the third component of tropomyosin binds to calcium. You see where we're going here? Have you figured it out yet? Nerve signals the muscle. That electrical wave slides across the plasma membrane and down the T-tubule. That T-tubule is close to the sarcoplasmic reticulum that causes the release of calcium into our cytoplasm. Calcium binds to troponin. Troponin pulls tropomyosin out of the way, which allows the myosin head that already has the energy in it to bind to actin, and you get a contraction. Those are the steps of our muscle contraction. And you don't need to get those now. We'll get them Wednesday and Friday. But I want you to understand the significance of all of these things as it relates to the bigger picture. Okay? And that's why we're covering all of these little components right now, because they all are the gears and the cogs of the machine that's going to be a muscle contraction. So once again, let's go back for perspective. Here's our muscle fiber, which is the whole cell, right? Here's our myofibril. That's a whole collection of our filaments. And our filaments are going to be lined up in such a way that we have these alternating light bands and dark bands. Light bands and dark bands. You see those? You can even see them at the light microscopic level. Now remember, we said that 80% of the volume of a skeletal muscle cell are these myofibrils. These cells are so packed with myofibrils, the nuclei get squished out to the edge. Those are nuclei. They're not in the middle of these cells. There's no room for the nuclei, so they all get squished out to the side. But what I want you to understand when you look at this illustration, we've got our thick filaments, we have our thin filaments. And do you see how our thin filaments are arranged almost like the branch of a tree, with the trunk being this purple squiggly line? This purple line running right down through the midst of our thin filaments, that's called the Z disc or the Z line. And that Z disc or the Z line defines the boundary of our contractile unit of our myofibrils. And that contractile unit is called a sarcomere. A myomere is a chunk of muscle at the gross anatomical level. A sarcomere is the repeating contractile unit of our myofibrils that's composed of our thick and thin filaments. And so here you can see a sarcomere from that Z line to that Z line. There's another sarcomere between that Z line and that Z line. So during a contraction, all sarcomeres shorten. And they shorten. And they shorten. All at the same time inside the entire myofibril. All the myofibrils contracting inside the muscle fiber. All the muscle fibers contracting inside the entire muscle organ. That's what shortens the muscle. So there's our Z disc to Z disc. Now I want to look at the light and dark bands. Now, whenever something appears lighter or something appears darker, what's sort of a easy difference to think about? Let's do this, okay? You can't see in my coffee cup. Can I hold this up a second? Okay. What is that? It's water. It's water. Coffee cup. Good job. You can see through water, right? Light can pass through it pretty easily. Has anybody got a clear coffee cup? Okay, imagine that was coffee instead of water. Why isn't light passing through the coffee as well as it does the water? Why is it darker?
Because of the coffee in it.
Because of the coffee in it, the stuff in it it's more dense. There's more stuff in the coffee to keep the light from passing through. Water is just water, right? So when we look at our banding pattern, where we see lighter bands, that's going to be where there's less stuff and more light can pass through. And these lighter bands that we see that have the Z disc right in the middle of them, those are called the I bands. I stands for isotropic. You don't have to remember that. I like to think I bands, I can see light. It's passing through. And the reason light can pass through pretty easily is because you only have the thin filaments that are keeping the light out. Do you see how the I band stops right where the thick filament starts? So the I band, only thin actin is present in the I band. We have some other structural proteins. We're not going to worry about those. We're just focusing on the thin and thick filaments right now. Now the I band is the easiest one, because when you look at the dark band, you see how it's dark on the edges, but in the middle it's lighter? And then there's that line right down the middle. Let's start looking at the darkest part first. The darkest part of these banding patterns is going to have the most stuff. And the most stuff that you could have in a myofibril is where you have both the thick and the thin filaments. So do you see here on the edges that corresponds to these dark edges? That's going to be where you have both thick and thin filaments overlapping. So it's the most stuff. It's going to be the darkest of the bands. And now do you see why the middle of that is a little bit lighter? Because look, the middle, where the thin filaments stop, there's less stuff in the middle, and so it's going to be lighter. And that middle portion of the A band is referred to as the H zone. Whereas the edges of our A band are thick and thin, they're the darkest. The H zone in the middle is lighter because it's only the thick. And if you could compare how light the H zone is to the I band is still going to be lighter because that's only thin and this is only thick. But it appears lighter because it's bounded on either side by the darkest regions of our overlapping thick and thin. Now finally, right down the middle, the anchoring line, you've got structural proteins that hold all these thick filaments in place and in alignment. And those structural proteins are called the M line. That's my body in the illustration. I'm the N line, and then here's those myosin molecules that are just waiting to grab the actin and slide the actin toward the middle, the M line, to shorten our sarcomeres. And when every sarcomere shortens, that shortens the entire muscle. Any questions about the banding pattern, questions about thin versus thick filaments?
So the A band is the darker band?
Okay. The A band is going to be comprised of the region that is defined by your thick filament. The I band is the region combined by a portion of your thin. Because do you see how a portion of the thin filament sticks into the A there on the edges? So see, your A band is going to start right at the edge of that thick filament. But that thin filament comes into just a little bit of the A. That's going to give you that dark region on either side with the lighter H zone in the middle and that dark line right down the middle being the M. Yes, ma'am.
So the H zone and the M line are in the A band?
Correct. Did everybody hear that? That's a good way to put it. So the H zone and the M line are within the A band. The I band is easy. It's only actin. But the A band has three different regions. The sides, which are both thick and thin filaments, The H zone, which is only thick, and the M line, that's the thick filament, and these other structural proteins that make it look a little bit darker right down the middle. But all of that is within the A band.